So JJ and I will be presenting fluorescence in situ hybridization, or fish. So what is fish? It's not an animal. It's actually a way to visualize and map the genetic material in individual cells, and this includes genes or segments of genes in the cell on the chromosomes, and it's important for understanding a variety of chromosomal abnormalities and other genetic mutations, which we'll get into later. And one of the biggest reasons that FISH is a very useful procedure is that it's much more versatile than the commonly used procedures, because the cells don't have to be actively dividing.
First discovered by biomedical researchers around 1980, and since then they've made a lot of improvements in the labeling of the DNA probes, which allow the hybrid probes to carry enough fluorescent molecules for the direct detection of the chromosomes. So that pretty much means that earlier, when they first discovered it, you could barely see anything, and now the fluorescence is it really stands out. You can see it right away. So FISH is a cytogenetic technique. A little bit about cytogenetics. It is the study of inheritance in relation to the structure and function of chromosomes, which is very relevant because FISH is entirely chromosomes. Cytogenetics mainly works with G-banded chromosomes, which means they're really good for karyotyping. And the other technique that they typically use is comparative genomic hybridization.
How does FISH work? Basically, FISH is very useful in helping a researcher determine where a gene is located on a chromosome, or multiple genes, because you can tag for different genes on a chromosome. It's originally performed during metaphase of a cell, but recently it's been able to be performed on cells in interphase, which has huge implications in cancer research. And also, FISH is more commonly used now because it's a less labor-intensive method for mapping the entire genome, which would typically have been done with southern blot. Okay, so like we mentioned before, FISH is used to detect the physical location of a gene on a chromosome. And FISH, fluorescence in situ hybridization, in situ actually means in place in Latin, and so most of the process is used, or is completed by actually affixing the cells in place on a slide, and this also affixes the chromosomes. So first what's going to happen during this procedure is that the cells are going to be treated to make them swell, which hopefully, yeah, so the cells get treated to make them swell, like this, and then the chromosomes are treated so that they denature into single-stranded DNA, which you can see here. So once it's denatured, it's ready for the next step of FISH, which would be to flood the slide with DNA probes. And these probes are chemically modified so that they specifically line up and hybridize with a segment of DNA which would have the gene, like you can see here. So these probes are also modified so that, they're also modified so that fluorescent probes can attach to them later. So the probes are washed away, and now fluorescent probes are flooded onto the slide, which you will see in 2 seconds, and they attach to anywhere the DNA probes had attached previously. So then those fluorescent probes get washed away, and now the slide is completely ready to be viewed under a fluorescent microscope, and so you can physically see where your gene is located on the chromosome. So this is the next slide. Basically, it's just showing what the video went over. You can see the DNA here, and then the probe DNA with immunofluorescent dye in this case is hybridized to the DNA at the site of the gene, the complementary DNA, and then you can visualize it on the microscope, fluorescent microscope. What is FISH used for? There are 3 different types of probes that scientists use. They are locus-specific probes, alphoid or centromeric repeat probes, and whole chromosome probes. Locus-specific probes bind to a particular region on the chromosome, and they're very useful for identifying isolated small portions of the chromosome. And they're used when a scientist wants to determine where on the chromosome a gene is located. Alphoid or centromeric repeat probes are generated from repetitive sequences towards the middle of the DNA, middle of each chromosome, and they're used to determine whether an individual is missing genetic material on that particular chromosome. And then lastly, whole chromosome probes are actually a collection of many different types of probes with immunofluorescent tags, and they each bind to a specific sequence of DNA, so each chromosome gets its own unique color. This is called a spectral karyotype, which you can see in the lower right corner, and this is particularly useful when you're examining for chromosomal abnormalities, like when one leg of the chromosome attaches itself to a different chromosomal leg. And also, it's useful for looking at aneuploidies in different cells.
So fish can be used for a lot of really cool things. It's got some pretty sweet applications. So the first one is for species identification. So let's say you're a patient. You're diagnosed with this pathogen. You don't know what it is. It looks like that stuff over there. You're not really sure what's going on. So they can take bacteria from your tissues or your fluids and grow it up in an agar, and then they can use fish to identify the pathogen. It's pretty cool. So it's also really prevalent in evolution. So they compare the genomes of two biological species to prove like evolutionary relationships, like where it was and how it's related to other things. So it's really similar to a zooblot, which I have pictured here, except in fish, when they do it, they use a karyotype and they compare the chromosomes rather than comparing the DNA fragments in the zooblot. Alright, so another scenario, let's say that you're a parent and your child has a developmental disability. But you don't know if you want to have more kids or not, because you don't want them to get the disability too. So you can sequence your DNA and your child's DNA, and then if they still don't know what the issue is, they can use fish to make the karyotype. And they can use that to test for Prader-Willi syndrome, Angelman syndrome, and chronic myelogenous leukemia. It's a big one, as well as many others. Here's our references.